Good morning, everybody. Today we have our final lecture of 5 for the Bioinformatics unit in the honors year. It's been a lot of fun working with you guys. Thank you very much for all the questions and feedback you've given me on the lectures. This has been really great. So today we are talking about biological pathways and networks. And I don't care who you are, at some point you're going to need the ability to evaluate pathway and network information from biology experiments. So I hope that this lecture gives you some pointers on how to get started with that. We will begin by a discussion of why organizing our data at the gene set level or the pathway level, for example, is a viable strategy, one that is very helpful for us in understanding the meaning of our data. We will talk about several different kind of constructs that we use, pathways, the Go categories. Most people, I think, will have heard of Go categories without too much delay in grad school. So Gene Ontology will be one of these systems that we evaluate. We'll talk about network modules very briefly. The network biology aspects of this may fall in after the intermission, and I don't know that we get past the intermission for this lecture, so that may mean that we're cutting short the topics that we could possibly discuss. Sorry about that. But we'll talk about enrichment analysis methods, specifically the overrepresentation analysis. A lot of people assume that if they're doing pathways and networks, they've got to buy one of these expensive licenses to software that's commercially available for this. There's a famous package called IPA, for example. Ingenuity pathway analysis is kind of expensive to get a license to. But the good news is you don't actually have to have commercial software in order to do pathway analysis. Systems like WebGestalt are absolutely free. There's also a really famous toolkit called Gene Set Enrichment Analysis from Jill Nesarov over at MIT. That is a very widely used toolkit, especially in the case of interpreting microarray data. All right, so where do we get information about pathways and networks? We're no longer thinking in a gene-by-gene context. What does this gene do? What is its sequence? What are the modules we can annotate within it? We're now thinking about a pairwise exploration or a group-wise exploration of the genome. What do these genes have in common? What are the inputs to this pathway? What are the outputs to this pathway? What are the intermediates along the way? And so we're moving out of a space where it's a gene-by-gene assessment and thinking about them as collectives. That's going to give us a lot of differences. And one of the key things that this ties to is the concept of relationships, that this gene and this gene are not independent of each other. If they're part of the same pathway and this one is upregulated, that's an indication that the pathway may be upregulated. These are no longer independent. They're both up and both down together. We frequently find that kind of correlation of expression tied to relationships among different genes. But there are a large number of ways that we get these relationships out of genes. So we start with just classical biochemistry. If you flip through your biochemistry textbook and find the Krebs cycle, you see somebody who whiled away his entire career on just how the central metabolism of cells works, right? So classical biochemistry has given us a lot of pathways that say this gene hands off its product to this gene hands off its product to the next. So biochemistry textbooks are full of these, and these pathways and network databases give us a way to codify that information so we can use it computationally. Microarray correlation is a very big one. I mentioned an example just a moment ago. If you have multiple genes that are in the same pathway and you find that these two genes are both expressed prominently in the same conditions that their correlation values correlate across different states of excitation for the cells, that's an indication that they may be involved in the same process. So implicit functional relationships are built from the idea that these genes come on and off under the same conditions. Chromatin immunoprecipitation, I think many people here will have heard of chip analyses. This is one of the applications that we put modern sequencers through. Protein immunoprecipitation is another key example here. That you have an antibody against your favorite protein, you yank it out of solution and notice that it's bringing along hitchhikers. These are other proteins with which it interacts. So when we find relationships like these, we can say, if these two proteins interact directly, it's likely that they are involved in the same processes at least part of the time. So protein immunoprecipitation is very valuable for that. The yeast-2 hybrid system is a very widely used genetic method for determining the interaction of multiple genes. Confocal co-localization is another big one. A few weeks ago we had a lecture in the Friday mornings about the use of fluorescence microscopy, in this case not confocal, but in cases like this you might have a dye that a fluorescent dye that you attach to, that you engineer into one protein and another fluorescent dye that you engineer into another. If you can show that red and green are always in the same location making that yellow light, that's evidence of co-localization of these proteins as well. Again, we assume that if these two proteins are in the same location, they have to do with the same process. So these are the wet techniques that get us to the dry databases that say this gene and this gene are related on the basis of this evidence. We also frequently find controlled vocabularies come into play. Controlled vocabularies can be sort of a niche area, but they're actually quite important. A controlled vocabulary suggests, well, to give you the example of the Human Gene Naming Consortium, we long had this problem where different groups would use different names for the same gene. That led to a lot of confusion. So now the HGNC has specified exactly which names will be used for which genomic coordinates to relate to what genes. That's an example of a controlled vocabulary. But you can do the same in terms of the biological experiments that led to information about a gene association. So you may find that a controlled vocabulary is used to say why we think this gene and this gene are part of the same pathway. Okay, so controlled vocabularies plus relationships have led to things like gene ontology. One of the big heavy hitters in the space of pathways. We're going to have more slides on that in a minute. The canonical pathways have also been visualized and made concrete in a lot of different ways. We're going to talk about some of the many pathway databases that have been spawned to store this information. But increasingly, we've seen people moving to a point-to-point network system. So instead of saying these 20 genes are part of one pathway and treating them all as equally strongly associated with that pathway, we instead talk about the distance between genes. Maybe we know that A interacts with B, B interacts with C, and C interacts with D. So we could say from that, well, obviously A interacts with D as well, but it's actually doing so through multiple links. So the action is not as directly connected as a case where A interacts directly with B. Right? So we find that people are moving to the models of biological systems that are built on point-to-point networks, biological networks, rather than large-scale pathways. The second half of the lecture is a lot about the modeling that's required to get to that kind of system. So a lot of what we're going to talk about today is going to be about pathways rather than this point-to-point network system. There are lots of things that can result from this. Being able to use point-to-point, gene-to-gene interactions to say what are tightly connected modules, network modules within cells, is kind of an alternative to this pathway ideology. Random walk assessment can talk about how strongly related things are from different parts of the network. Remember we said A to B, B to C, C to D, and so on. That's an example where you would have 3 steps to get between this gene pair. That reflects that they are probably less related than things that are directly connected. Cytoscape, one of the most important tools in network biology, is certainly something that we'll want to talk about along the way. Dimensionality reduction is a very big piece of this, and it's something that we're going to come back to in just a bit. But as I mentioned, being able to say that this set of differences is enriched for a particular kind of activity is something that we routinely want to do with our pathway analysis. We're going to talk about 2 methods for that down here at the bottom. Okay, so where do we get our information from? One can easily separate, one typically needs to separate at least, a difference in recognition or identification and differentiation. So imagine that you are using a microarray. That microarray has some number of probes on it, probably better than 1 million, in a lot of cases better than 100,000. And we're able to say, out of all of the genes in the genome, how many do we have any detectable expression? So you might think of that as a limit of detection. You probably will want something better than detection, though, in order to say this set of genes is differential. Do you see why being able to say I have any signal for this probe is different than being able to use it statistically? Okay, so we would speak of that as a level of quantitation. So in a lot of the data that we generate from differential proteomics or differential gene expression, we want to be able to say these are the genes for which we have any information, and then within that we have a smaller set. This is the set of genes for which we're able to test a statistical hypothesis of difference between these two cohorts. So whether you're dealing with microarrays, or dealing with RNA-seq, or dealing with proteomes, you're quite likely to be performing differential experiments. We have this cohort and that cohort. This one was not stimulated, this one was stimulated. These are the genes that or proteins that appear to have different levels of expression. Let us then say that with this list of differences, we can then go gene by gene and put together a t-test for each gene. We saw plenty of examples of that with a practical on Friday, for example. We might also use clustering. I know we talked about clustering a couple slides back, but you might say that these genes all act in concert across these 50 different stimulations of the samples. We saw that these genes always rose and fell in concert with each other. So something like that could easily be handled through clustering. So let's imagine then that we have some list of genes that we've got our attention fixed upon. Maybe we measured some 10,000 genes with a microarray, that we were able to detect evidence from 10,000 genes. And we were able to do differences on some subset of that. From that, we've now got a list of, say, 35 genes or whatever. Come on. We now have a subset of genes drawn from those we detected that have putative differences. So this list has already come through quite a lot of steps to get to this stage. And now we see that these genes are of interest to us. How are we going to figure out how they relate to each other? So a lot of times we think of people enumerating genes and then putting together some sort of assessment of what types of activities are attributed to them. So I've just pulled just a few functions from a few very popular genes. So CASP up here is part of the MAP kinase pathway. CASP is part of the apoptosis pathway. RAS, another really, really popular gene for study, is part of the MAP kinase pathway as well, and cyclic AMP signaling. MEK1, it's also part of MAP kinase. Apoptosis is another follow-on for it. MLCP, also involved in cyclic AMP signaling. So we can think of it that way. For each gene, what are the possible activities in which it's involved? But you could just as easily turn this inside out. So let's transmogrify this list to say, not what pathways, what activities are associated with these genes, but rather what pathways help us to organize the data, which pathways are most popular among all of these genes of interest. So we see that of all of these genes, the MAP kinase one is rather popular. Only one of these genes, MLCP, was not associated with the MAP kinase activity. Apoptosis, it showed up a couple of times. That might be a reinforced area that we want to be looking at as well. So we've turned our list inside out. Instead of saying which functions are associated with these genes, we ask which genes are associated with the major functions present in these data sets. All right, so what is the value of pursuing this kind of assessment, which pathways are enriched among these among this gene set? We start with better interpretation. I want to remind you of Feynman's adage: no one is easier. You'll find no one easier to fool than yourself. Right? In the same way, if you give an experienced molecular biologist a list of 20 genes chosen at random with no biological basis whatsoever, just 20 genes chosen at random, they will come up with a hypothesis that explains how really this explains what they've known all along about the biology of cells. That's a very dangerous kind of habit, and we engage in it all the time. So if you want to improve the interpretability of your data, turning the data inside out in this way, saying which pathways are associated with lots of genes on this list, is a way to protect you against that. It gives you better interpretability to be able to say not just that these random genes seem to be differential, but it's far better if you can stitch together the pathway that spans over a large number of them. This also gives you improved robustness. So if you examine the data for each individual gene, the amount of information you've got to decide whether this gene is differential or not is less than if you were to look at all of the genes associated with a given pathway. Now you have a larger basis of information upon which to make that judgment. And this also leads to improved sensitivity, which is to say that if you recall, when we talked about differential gene expression, we mentioned the need for multiple testing correction. And the number of tests you perform is a very big factor in how multiple testing correction works. So if you can test fewer hypotheses, you've got better ability to accept a higher p-value as significant. So your chance of missing something because of multiple testing alone is already better, simply because we've got fewer hypotheses, fewer pathways than we have genes representing them. Okay, worlds and worlds of pathway databases are out there. A few years back, I went through this list and tried to update it with current URLs and so on. We had to strip some of them off the list because some of those databases had gone away. Now, if I do a search of all the pathway databases that have been published since I made this slide, well, it's going to get, the font sizes are going to get very small, because pathways and networks are a very active area of bioinformatic development. But you can see that all of these have viable URLs that you can pop off and start looking at. I've included an example here from BioCarta at the upper right, and down here at the bottom right, a service called KEGG. We'll be talking about KEGG more here in just a minute. But lots and lots of places out there have tried to assemble reliable pathway information into publicly usable databases. Many of them are free. Gene Ontology, however, is the big granddaddy that seems to get a huge amount of attention in this space. So it's relying upon a controlled vocabulary. It's structured, precisely defined for describing the roles of genes and gene products. And this controlled vocabulary is very frequently a part of an ontology. But who here feels really comfortable defining ontology? It's not a very usual word, is it? It actually comes quite heavily from philosophy. It's a theory about the nature of being, or the kinds of things that have existence. Ontology, that's what that term means. So if we're trying to create a controlled vocabulary about the things that have existence in genetic relationships, we're going to have, we need to define the nature of those relationships, and we need to define the particular activities that encapsulate what happens in biological systems. So you might consider something basic like a kinase. A kinase. Have people heard the term kinase? Okay, good. So a kinase does what? Very nice, yes. Yeah, kinases attach phosphorylations to other proteins, typically at serines, sometimes at threonines, sometimes at tyrosines. Okay, so a kinase is a type of biological activity. This is the sort of term that you would expect to have in a controlled vocabulary about molecular function, because a kinase typically operates as a protein that activates another, that sends it into motion. So must have that. Okay, now there are three different ways that things get sorted by the gene ontology, and I would really like everybody to remember this when they're analyzing it. People like me, we tend to focus on one of these. Maybe I care about molecular function and I don't really pay much attention to the others, but these three pieces are all quite important. Molecular function is kind of a tactical view of what happens to these biochemical compounds. Biological process tends to be more of a gestalt that describes at a larger level what the role this has in keeping the organism alive. And cellular component tends to be much more about localization. This is part of a rough ER, for example. Did you have a question? Okay, cool. All right. So we see then that something like dopamine receptor D2, the product of human gene DRD2, it could be described as having a dopamine receptor activity. That's a molecular function. It sits around waiting for dopamine to arrive, right? Biological process means that this is, it shows that this is useful in synaptic transmission. So when you have an axon and a dendrite and two nerve cells are interacting with each other, the receipt of dopamine is part of this synaptic transmission, the communication between nerve cells. Cellular components says, well, this is found in the plasma membrane. Not the mitochondrial membrane, not the nuclear membrane. This is found in the cellular membrane. Okay, and of course the relationships, like is a part of, has the part, regulates. These are the kind of relationships that get described. So on the one half, relationships. On the other half, controlled vocabularies. Together, these create the gene ontology, which is really 3 hierarchies for the price of 1. So we see then that these relationships are frequently shown as edges in a graph. We could have at the very highest level of the biological process hierarchy a node called biological process, very intuitively named, I think. That can be subdivided into at least single organism processes and cellular processes. Single organism cellular processes inherits from both of those. Both of those higher nodes lead to this. So we're building a hierarchy. I'm not showing you the full tree because the boxes would be minuscule on screen. I'm just showing you this one little slice out of it. We see that cell death is a type of single organism cellular process, and programmed cell death is a particular subvariety of cell death. So we have this hierarchy all hanging down from a very generic to something very specific. And these relationships are spelled out by the colors that they're showing on each edge. You could also drop down from plasma membrane. We see intrinsic components of plasma membranes, extrinsic components, inside and outsides of membranes. We have things like intrinsic component of the external side of the plasma membrane, from which we have things like integral component of external side of plasma membrane. More and more specific, this time under localization. So, I'm sorry, cellular component is what that one's called. I always think of it as localization, so. Okay, so these are the kinds of relationships we have among different kinds of biological processes and different types of cellular components. All right. So we find that there are many, many ways that information get into the gene ontology. Electronic annotation is really handy, of course. But generally speaking, people feel more comfortable if a human has come along at some point to evaluate that information. So human annotators plus a bunch of algorithmic inputs give us this flow of information that makes Go better and better every year. All annotations must be attributed to a source. So if you're adding new information to it, you have an exact record of how that type of information got entered, and indicate what evidence was found to support the Go term, what type of gene-protein association was used to create this entry. We have evidence codes like IDA, IMP, IJI, IGI, IPI, IEP. You can look up what each of these codes means. We have computational codes, things that refer to automatic annotation that got dumped into Go. Sometimes we have author statements, right? That this professor just published a paper about this, sent an email to Go saying, my paper shows unequivocally that such and such gene is part of this pathway, therefore you must have that annotation. Well, that comes along with an author statement code in the database. Sometimes we get situations where no one really knows, and by mistake, information was added into Go, and someone simply writes no data on how it got there. That's always a little awkward, though. We try to resist that. Lots of places where you can do this. You might just go to the Gene Ontology website. That's a very reasonable thing to do, but everybody's made some sort of interface to interacting with it. So Amigo 2 is one that's offered by Gene Ontology. If you're using R, you'll find that Bioconductor has a really handy set of libraries, frankly, that can all speak Gene Ontology. And QuickGo is a service that's available from the European Bioinformatics Institute. So great stuff here. Now, do all organisms get equal amounts of annotation in Go? A definitely big no on that one. I, for one, love my house cat, and I really wish that we had a lot more biological information to go on when we were studying cats. The cat genome was sequenced many years ago now, and yet the amount of network and pathway information that we know that's specific to house cats is very sad. It's very sad. So when we look at our list of who's well covered, we can say, well, gosh, Homo sapiens gets an awful lot of attention. Mouse gets an awful lot of attention. Rattus norvegicus, everyone knows that one, I think. Rat, yeah, lab rats. Why Norwegian rats, huh? Maybe the Finnish rats need some love too, but that's all right. All right, Norwegian rats. How about Gaius gaius? Anyone have that one?
Chicken.
Very nice, the chicken, very good. How about Dana Rerio?
Those little things that live in water.
The little things in water.
Daphnia.
No, wait, actually, no, it's not.
Yeah, I heard Dana and then also.
I've read it.
I'm thinking that zebrafish, right?
Yeah,
Oh, yeah.
Dane is any little.
Oh, it lives in water, hey guys.
A little thing in water. Octopi? I don't know. Okay. Okay, Drosophila melanogaster. Fruit fly, yeah, pretty well studied. And C. elegans.
Is a flatworm.
Yeah, a little worm. Little worm. Okay, so one of the best developmental models. So we can see that depending on what organism you're working with, the amount of gene ontology information may be quite a lot or relatively little. We can also look at what kinds of information led to these being entered. Remember that we have 3 different hierarchies for each. We have P, F, and C, so cellular component, molecular function, and biological process. And in each case, we're asking for just human, these first 3 stripes are all human, what fraction of that information came from experimental evidence? I mean, we don't experiment with humans a lot, right? Try not to anyway. Curator inference, author statements, combinatorial evidence, etc. All of that, we can see how much of each of these has contributed to our knowledge of the biological systems in each. All right, now we're moving away from gene ontology for just a moment. This is a diagram from KEGG, the Kyoto Encyclopedia of Genes and Genomes. Very useful resource, but they have a whole lot of these hand-drawn maps, and I think it's kind of an interesting way to visualize this information. So in this case, we have our favorite molecule over in the green box. Can anyone read the tiny print?
Caffeine.
That's caffeine, right. So there's a whole biosynthetic pathway built around caffeine, and we can see that things like 1,3,7-trimethyluric acid can be generated starting with caffeine as a molecule. So your morning tea may already be on its way down that pipeline. This is a metabolite-centric network. So when we look at the individual nodes, these are not genes. These are metabolites, like caffeine. Caffeine's not a gene, it's a small chemical. So we can see then that there are relationships drawn between different nodes here, these stopping points, that have these little boxes with 4 numbers on them. Okay, so these boxes with 4 numbers in them are not IP addresses, right? I mean, they might look like 192, 168, 100 or something, but that's not what they are. These are Enzyme Commission codes. Has anyone heard of Enzyme Commission codes? Very valuable. When we try to say this activity in this species is handled by this gene, and that same activity occurs in another species and it's handled by that gene, we can relate these 2 genes together by saying these should have the same Enzyme Commission code because they represent the same activity in multiple species. Remember, we referred to those as orthologs before. That's usually on the basis of sequence similarity. On functional similarity, we would make the decision that these enzymes have the same Enzyme Commission code. So when you see these dotted quads of 4 numbers, those are representing Enzyme Commission codes, one of the earliest ways to try to store pathway information. Okay, which brings us to WebGestalt. So we've talked a little about gene ontology, we've talked a little bit about KEGG. These are canonical pathway systems. This is one of the best workbenches for dealing with it. So we start with the fact that you have experimental data of some sort. In this case, we're using microarray probe numbers. Okay, so we have these microarray probe numbers that we want to analyze by a pathway analysis. These flow into the system, and we need to make some sort of mapping from our probe numbers to things like a KEGG overlay. And from that, we see that we're going to have some sort of translation capability to something that knows what this probe number means in relation to gene symbols for this species. We can see that there's quite a few that are available to us. Just in microarrays, we have Affymetrix arrays, Agilent arrays, code-link arrays, Illumina arrays. All of these use different addresses to specify what each probe on the microarray means. So the ability to translate those to genes is one of the first things that WebGestalt does. One of the features that I really, really love about WebGestalt is that it tells you how successful that step was. How would you feel if you handed in, let us say, 100 probe IDs from your microarray representing differential probes. And the software said, I can only identify gene names for 50% of those. That would be a matter of concern, right? But a lot of the software we use for pathways and networks assumes that that step just went fine and doesn't bother telling you what fraction of the probes were simply not recognized. WebGestalt makes that process much more transparent. So it's one of the nicest features of it. You can see that it has a wide variety of species and identification types available to it, and it offers quite a lot of functionality for performing statistical analyses on the data that pass into it. Okay, and just along the way here you can see that under pathways, it includes KEGG, it includes pathway commons, it includes wiki pathways. Gene Ontology is listed as its own separate box. Network modules, this group has been one of the most heavily involved in finding out point-to-point descriptions of biological activity. Disease-associated genes, drug associations, all of that stuff is already made accessible within this web framework. So over-representation analysis is one of the types of analysis that are most commonly used in the context of pathways and networks. So I want you to imagine that we have data that came from our microarrays, and the microarrays in this case gave us more than 500 genes that it claims are differential. You know, this one's like that, this one's like this one has a p-value that puts it way up here on the volcano chart, this one has a p-value that puts it much lower on the chart. So these represent your experimental data in yellow. At the left in green, we have information from something like Gene Ontology. So in this case, these are genes associated with development. It's a pretty big category, but that's what we've got. So 1,842 genes that are known from all of the experiments that we've done in the past that are part of Gene Ontology's development category. So if we have this list of genes that we've detected as differential, how can we tell whether there are more or fewer than we would have expected by random chance in development? That's a little tougher. So I want to point out that there are lots of ways that these can intersect. If you were to draw these 581 genes at random from all detected genes, you would expect there to have been 65 that were shared between these lists. But in our case, we didn't see 65, we saw 98. So is that enough to get excited about? Okay, so let's start with the expectation. What if we saw exactly 65 genes that intersected between these two lists? That's exactly what you would expect by random chance alone. That's no vote in favor that development is a major player in whatever is causing this difference, right? So the higher this number gets, the further we get upward. The best possible result here is 581, right? That every last one of our differential genes fall in a particular pathway. That's brilliant. But in this case, we've got something more moderate. 98 genes overlapped between development and our list of differential genes. We need to have some way to put a probability on that. So one of the most common approaches that we use is built on the hypergeometric distribution. The hypergeometric distribution lets us think of all of this in terms of a contingency table. So we have genes that we found to be differential on the left, genes that we did not find to be differential on the right. We have genes that are in the development group on the top row, and genes that fall in other groups instead on the bottom row. All of that added up is going to give us the ability to power a test like this. So we saw the hypergeometric distribution once before. Do you remember my discussion about black and white marbles and trying to match tandem mass spectra? It's the same model, just being applied in a different context. So in this case, we're asking, what is the probability that we would see 98 proteins in this overlap by random chance? What is the probability that by random chance we would see 99 proteins in this overlap? What is the probability that we would see 100 genes in this overlap? What is the probability of 101? And keep going all the way up to 581. Those probabilities then get summed together. Remember, we've got this big sigma symbol here. That's the summation of all of those probabilities. So in effect, what this model is telling us is what is the probability that by random chance alone, you would have an overlap this big or bigger if you just had random gene draws. Okay, so this gives us the ability to say, to put a probability on it, that we do not think that by random chance alone you would have this strong an overlap because of the probabilities that come out of this. Okay, so in the example that you can run right on the WebGestalt website, they have a tutorial data that you can apply here. If you use this, we see enrichment ratios are one of the outputs that come from this, and we've got false discovery rates because we have a lot of different pathways that we're testing for their overlaps with our experimentally derived difference set. So what we see is that the enrichment ratio that's highest is associated with the peptidyl serine modification. The information that leads to that is visualized down below. So we can see that we had a gene set that was 305. The number of genes associated with peptidyl serine modification was 446. And there was this overlap of 35 that was much, much more than you would have expected to happen by random chance alone, which is what pushed it all the way to the top. So this is valuable to you because now instead of talking about this gene and the other gene, you're talking about function. And function is very frequently a much easier place to start talking about the biology of your samples, not just about individual molecules that play a role in that. You can also do your projections, though, in things like KEGG pathways. So here you can see that just a very few of these genes are in blue. Those are genes that were part of this difference list. Our analysis said these genes are differential. The code then has drawn this map with coloration to reflect those genes that were part of our difference list. This is why we can feel much more strongly about saying this pathway is involved. And you can use this for some error checking too. What if you were looking at CTN, NB1, and LEF1, both in this chart, and you saw that the first gene was way up and the other gene was way down? Wouldn't that be kind of confusing? We would expect those functions to be correlated, so if one is up, the other is up. So they could reinforce each other, or they could countermand each other. Okay, great. You can also do this, as I mentioned, in a point-to-point network. So here we are drawing sets of genes that have edges drawn between them because that pairing of genes has been seen to be, for example, to form a heteroduplex or to be correlated with each other in microarrays or whatever. So these are not all together because they've been called a pathway. These are all together because each of these interrelationships between pairs of genes is strong enough that we can, when we pull one out, we kind of pull out the whole mess with it. So this kind of network module approach is another very fine way to describe these associations. Okay, so there are some limitations, obviously, to doing it this way. The first one, I would say, is that we have done a fair bit to protect ourselves against multiple testing correction in evaluating gene-by-gene data. You know, when you deal with a microarray, we have pretty good systems for limiting the errors that we're going to see. When we make GWAS studies, we have pretty good strategies to figure out which markers are acceptably differential in a population. But I would say that we still have some problems to tackle as we deal with pathways. Remember that a lot of this pathway information is hierarchical. So you can imagine then that the genes at this point in the hierarchy tend to be a superset of all the genes in the box hanging off of it. And that in turn is a superset of all of the genes hanging off of that. This creates a problem that the p-values that relate to those different stages of the hierarchy are correlated with each other. We cannot treat these as independent p-values. So that can make understanding these data a little challenging. Also, if you are to say that all genes that are associated with a given pathway have an equally strong association with that pathway, that's also a little problematic. You know, if you're trying to show caffeine metabolism, a gene that interacts directly with caffeine by producing it or by using it as an input is much more strongly associated than something that's 3 or 4 or 5 different catalyzed reactions away from it. So we have this problem that some of the data are some of the genes are much more strongly associated with this activity than are others. So treating pathway genes as a set ignores that some genes are central to those pathways while others are quite tangential. So Gene Set Enrichment Analysis has really proven to be quite helpful as we try to evaluate microarray data. People are coming to realize that it's quite useful in lots of gene expression studies, not just those for microarrays. I find that explaining how it does its thing is a little challenging, but let's try to work our way through this gradually. First off, we're accustomed to saying that this set of genes is differential up, and this set of genes is differential down, and then there's this great mass of genes that we're saying have no differences. We're going to have to get out of here in 5 minutes. Sorry about that. So if you have your genes sorted in this way, you have those that have significant p-values and are way up, those that have significant p-values and are way down, and then things that have p-values that are quite indifferent, that range from a little up to a little down. So we can sort them all on that axis. Now, if you can add that these genes all have particular pathways to which they're associated, you can go pathway by pathway to ask, do I find a whole bunch of genes associated with this pathway near the top of that list or near the bottom of that list? And you can think then of this expectation score, the enrichment score sort of rising a whole lot as you encounter many, many genes that all hit that pathway, and then sort of fading as you get into this gray stuff in the middle of our list that doesn't have significant p-values. So the best scores that we get on pathways are going to be from things that have a whole bunch of hits clustered together at very low p-values and up, or things that have a bunch of very low p-values that are down in fold change. But it's non-parametric in the sense that it doesn't care about the magnitude of these p-values anymore. We're not saying these genes are differential and these genes are not. We're saying that the evidence for these being differential is greatest, the evidence for these being differential is quite minimal, and once again, as we get to the other end of fold change, we see that these others also have a greater chance of being differential. So Gene Set Enrichment Analysis doesn't imply these really hard thresholds that we usually associate with overrepresentation analysis. Okay, so in pathway-based assessment, we want to organize our genes by either pathways or maybe by gene ontology. We have enrichment analysis like overrepresentation and gene set enrichment analysis that have greatly helped us to understand the biology implied by these samples. But we always run against this problem that our annotation of function with genes is always woefully incomplete. What do you do when you have different pathways that say there are only 5 genes associated with this activity in wiki pathways, but 8 in gene ontology? That's kind of awkward, right? So the information in pathway databases is never comprehensive, and it never agrees perfectly. So what you get out of your analysis will probably reflect, at least in part, what pathway you're using to interpret it. That brings us to the intermission. Because we have only the hour today, we are going to call this to a halt. But we will be back in this room at 1 o'clock today. We will have 1 hour to do our demonstration this afternoon. Please be here make sure you're here by 1 o'clock because Sam Sampson is going to come by to talk about the I'm not going to put you on the docket to speak. Sam Sampson is going to talk about the mycobacteriology advanced track. And we want to make sure everyone is here for that. So thank you very much. I'll see you this afternoon.